Good afternoon, everyone. We are going to start our presentation. His name is Dr. Enrique Salas from Instituto de Biotecnologia from Universidad Nacional Autónoma do México, and he is going to present zebrafish as a model for the study of reactive oxygen species functions in animal development. He got his BSc degree in biology at the Nacional University of Mexico and a master's degree and PhD in biomedical sciences at UNAM. He started to work with zebrafish as a model organism during his postdoctoral research work at Leiden University in the Netherlands with Professor Herman Spank. He is currently a full-time professor at the Biotechnology Institute, one of the major research institutions in Mexico that belongs to the UNAM. The goal of his current research work is to understand the functions performed by reactive oxygen to the control of early animal development processes and how they interact with the non-molecular networks that regulate early development. As a main experimental model, he used zebrafish embryos during early cleavage up to late gastrulation. Thank you, Dr. Enrique, to be here with us and have a good presentation.
Thank you very much. First of all, I thank you, Monica. I want to thank the organizers of the meeting for inviting me to participate here. I hope you can see clearly the presentation. Yep. Well, I will start. So we use zebrafish as a model for the study of reactive oxygen species functions in animal development. And okay, we'll try to move this thing. And actually, reactive oxygen species are generated through the partial reduction of molecular oxygen. There are different molecules, but the most known or best characterized are superoxide and hydrogen peroxide. And in particular, we are more interested in hydrogen peroxide because it's known that it participates in the regulation of different fundamental cell behaviors, like in the control of cell death, of proliferation, of differentiation, and also in the control of motility and migration of cells. And all these cell behaviors actually are important for normal development of animals to take place. And well, how do we detect ROS in tissues and organs, or even in complete embryos undergoing development? There are many kinds of molecules that are used as ROS sensors, but in particular, we have used for several years DCF. And this is a compound, and there are many derivatives of this compound, the dichlorohydrofluorescein diacetate, that actually is non-fluorescent. It can enter into the cells, or it can enter into the different tissues and organs, or even in complete organisms. And there, in different compartments, actually is processed by esterases, and is maintained in those different compartments in the embryo. So it can undergo oxidation by the activity of different ROS. And with this molecule, we can detect regions that are undergoing oxidative stress, because the compound that is generated is a DCF, that is a fluorescent compound. So now, since it is fluorescent, we can detect it by conventional fluorescent microscopy, or by different techniques like confocal microscopy. So reactive oxygen species are known to be required for different process of morphogenesis. And when I was doing my PhD project many years ago, actually, I was working with a very nice morphogenetic process that takes place during limb development in the mouse. This process also actually occurs in humans. At the beginning of development of the mouse palm in the limb, actually looks like a palette, and the fingers separate among them, thanks to the regression of the interdigital tissue, because there's a quite a large amounts of cell death that occurs in that tissue. At that time, we found by using this compound, the DCF, that ROS accumulate exactly in large amounts at the interdigital tissue. And when we block the ROS levels, we downregulate them by the use of different antioxidant compounds. We see that the cell death is downregulated, but also we affect the regression of the interdigital tissue, and so no longer the fingers are separated in this tissue culture conditions. At that time, also, I was very interested in trying to find out the patterns of cell death that occurs during normal development in mouse, and also to try to find out whether ROS are accumulated at those regions where there's a huge amounts of cell death, and if ROS actually participate in the control of cell death in those tissues. However, very soon I found that obviously mouse embryos are large embryos, and they are also not very transparent. They are quite opaque. So we were able only to look at the most superficial layers of the embryos. But I was very fortunate to start working with the zebrafish early development when I did my postdoctoral work in the Netherlands at Professor Herman Spank Laboratory in the Leiden University. And I will talk just briefly about the early zebrafish development. Once the egg is fertilized, cytoplasmic currents form the first cell on top of the yolk. This actually, the yolk is very important because it allows the embryo to develop outside the mother. And these first cells start undergoing cell cycles, and about 10 cell cycles are more or less synchronized. And these cells are sit on top of the yolk. So when they reach more or less a stage that is not shown here, actually, the 1,000 cell stage, some of the blastomeres fuse with the yolk, and then they liberate all the content, the cytoplasmic content, and also the nuclei into the yolk, and now the yolk start to have again nuclei. Yeah. And the cells of the blastodem that are sitting on top of the yolk, that after 4 hours of after fertilization, they reach a stage that is known as a sphere, because the embryo have this shape of a sphere at that time. But they start for the first time showing very dramatic cell movements that are part of the gastrulation process. And the first cell movement that is massive is a process known as epibole. During this process, actually, the cells of the blastoderm thin and spread over the yolk. And this can be seen in this beautiful movie from Kallstrom and Kane from 1996. So you can see that how the blastoderm cells thin and spread over the yolk and until it's completely covered. And in about 24 to 30 hours, you can already recognize an embryo and different structures like the eye, the brain, and the tail is already quite well formed by 24 to 30 hours of development. During early epibole, the embryos have 3 different cells types. So the deep cells are at the deep cell layer, and these are still very active, mitotically active, so they divide quite a lot. And these are the cells that actually contribute to the formation of the embryo and to the adult tissues. The other cell type that is found at these stages of development that is part of the blastoderm tissue are the enveloping layer cells. These cells also undergo the process of epibole, but they stop dividing more or less around this stage of development. But the epibole that occurs in these cells because they flatten, as is represented in this cartoon, and they spread over the yolk. And the yolk is very interesting because the nuclei of the syncytium actually lead the whole process of epibole. So yeah, so they are in the front end of epibole process during early gastrulation in the zebrafish embryos. Gastrulation actually is a fundamental process for animal development, because thanks to this process, the germ cell layers are formed: ectoderm, mesoderm, and endoderm. And these two germ layers are important for the formation of the different tissues and the organs and systems of the embryo and the adult organisms. But also, gastrulation is fundamental for axis formation. It determines, it participates in the determination of the anterior-posterior axis, dorsoventral axis, but also by the end of gastrulation and epibole in particular, the mechanisms that start to determine the asymmetry, right-left asymmetry axis, start to become important during those stages of development. And actually, during gastrulation, different cell movements are occurring simultaneously, and these different cell movements are represented here by arrows of different colors at different stages of epibole and gastrulation. We are mainly focused on epibole because it's a very tractable process of massive cell movements. So when we started with this project, actually these were started as control experiments, but they became one of the main projects of in which we work with. We asked when and where are ROS generated during zebrafish development. And at that time, Mario Mendieta was doing his master's degree, and he was changing to start his PhD project. And he stained embryos with this compound, DCF, to look at where are ROS generated in zebrafish embryos. And I will show you here, this is Mario Mendieta now, he's doing his postdoc work in Singapore. But when he stained embryos at shield stage, here you can see the shield that marks the dorsal side of the embryos, and the yolk is covered more or less just 50% of its surface. The embryos actually shown very interesting pattern of staining at the blastoderm. But it's important to highlight that the yolk remained negative during the whole process of epibole. But it was very interesting to find a region with the shape of a ring at the front end of epibole that was quite positive for ROS formation. And we wanted to know, when we analyze it actually in more detail with higher magnification imaging, that the region that accumulates most of or more intense signal of DCF, of ROS accumulation, is the extracellular space, the interstitial space that is among the blastomeres of the deep cell layer. Still, the cells also show some degree of signal of ROS formation, but this signal is much less intense when it is compared with the extracellular space signal. We wanted to know if during the whole process of epibole, ROS are generated, and to know the dynamics of ROS at the blastoderm tissues. And also we wanted to know if this DCF ring that highlight where ROS are generated remain there at the front end of epibole. So in this, Mario did time-lapse imaging by confocal microscopy, and here we were able to see a very nice and interesting pattern of dynamics of ROS formation in the blastoderm, but the ring, ROS ring, is leading the whole process of epibole. So we wanted also to know where do these ROS come from, and we focus mainly on NOx enzymes because these enzymes are known to use oxygen, molecular oxygen, and they partially reduce actually this oxygen to generate superoxide. And this superoxide is liberated into the extracellular space, or the interstitial space. That is actually where we see the highest signal of the DCF compound when we stain whole zebrafish embryos. The superoxide can be dismutated into hydrogen peroxide. This can be actually occur spontaneously, but also superoxide dismutase activity that is located at the extracellular space can perform this conversion of superoxide into hydrogen peroxide. And hydrogen peroxide can go into the cells, to the cytoplasm, thanks to aquaporins that are proteins that are located at the plasma membranes of cells. We became interested in NOx proteins because, well, they were started by several groups, but in 1999, Lambeth group in the United States showed that NOx proteins, NOx genes are expressed in most tissues, most adult tissues in mice, but also they found that the embryonic tissues at different stages and different tissues actually express different NOx genes. Later, this was a very nice paper from Needhammer in 2009. He showed that, well, different NOxes are expressed in zebrafish, and but zebrafish only have 5 different NOx genes. NOx 1, 2, and 4. Humans, we have a NOx 3, but this gene is not present in zebrafish. And also zebrafish have NOx 5 and duox 1. Humans, we have another duox that is called duox 2. He actually, Needhammer found that when he exposed zebrafish embryos, when they wound the tail of the zebrafish larvae around, I think it was 3 days after fertilization, he could inhibit with the NOx activity with the general NOx inhibitor, the VAS 2870. Yeah, so we ask if in the system that we work with, we are able to decrease the ROS formation by treating the embryos with this inhibitor. So we focus at the front end of epibole. As you can see, this square highlight the region that we amplified in this image. When we treat the embryos with the control media, you can see here the deep cell layer and just a little bit a piece of the yolk cell that is at the bottom of this image. But you can see that they show a normal pattern of localization of ROS with this DCF compound. When we treated the embryos with different concentrations of the NOx general inhibitor, Mari was able to see that the signal of the extracellular space and the ROS ring that is at the epibole front, the signal is downregulated, significantly downregulated. But as we increase the concentration of the general NOx inhibitor, he found that also the cells start to show less intensity in the signal of the blastodem cells. And this is, yes, what this was quantified by Mario Mendieta. Yeah. So next, we wanted to know. Well, actually, these results show that NOx enzymes are active during these very early stages of development of the zebrafish, but also they are responsible for the formation of reactive oxygen species that are accumulating in the blastodem tissue. And we can detect this by the use of DCF compound. But next, we wanted to know if we downregulate ROS formation in zebrafish embryos that will start to this process of epibole, the process is affected. Then Mario started to incubate embryos at the sphere stage. That is just the stage before the start of epibole cell migration. And the advantage of this is that we can stop the embryo development and fix the embryos at the desired stage. And in particular, here we stop the embryos when they reach it more or less about 70 to 80% of epibole. So we stain the embryos to look at DNA, and with a cytox screen, we could detect all the nuclei from all the cells out and nuclei from the yolk also. But with when we stain embryos with phalloiding Alexa, we can more clearly see the front end of epibole of the deep cell layer and also of the enveloping layer that are advancing by epibole. We could also detect the front end of epibole of Jolk nuclei by the staining of the DNA. When we exposed embryos with the general NOx inhibitor, we found that there's a delay in epibole, but this effect was most clearly found in the deep cell layer. And as we increase the concentration of the general NOx inhibitor, we found that the enveloping layer also was starting to show a delay in epibole. Yeah, and this was significant. However, the nuclei from the Jolk didn't show any delay in epibole, even at these higher concentrations of treatments with the general NOx inhibitor. However, we wanted to know if these effects were specific, and with the treatment with the general NOx inhibitor, we were mainly affecting NOx and enzymatic activity. We have shown earlier that, we downregulate the formation of ROS, endogenous ROS formation, but we wanted to know if we add externally hydrogen peroxide to the tissue culture system, we can rescue the effects that are due to the effect of general NOx activity inhibition, and that's what we found. At that time, Mario Mendieta let the embryos develop up to 24 hours of development, and these are the control embryos that in these conditions actually develop perfectly well. Most of the embryos actually survive up to 24 hours of development. They show the eye, the brain, different brain regions, and the tail is extended in these embryos. However, when we treat the embryos constantly with this NOx inhibitor, embryos die after 24 hours in these conditions. Actually, Mario found that embryos stop epibole more or less around 80 to 90% epibole, sometimes even earlier at 70% of epibole. They freeze at that stage for some minutes or sometimes hours, but then during night, these embryos are incubated in constant concentration of this inhibitor. They die by after 24 hours of treatment with this compound. Mario was able to show that when he treats the embryos also with hydrogen peroxide in the media, he found that the survival of the embryos is fully rescued, and they also develop perfectly, and these embryos were undistinguishable from when we compared with the control embryos. And Javier Mendez Cruz, when he was doing his master degree thesis research work, he also shown that hydrogen peroxide treatment actually rescue the epibole delay that is provoked by the treatment with the general NOx inhibitor. And Javier also did very nice experiments that were the final experiments that we did for publishing this nice article some years ago, because when he treated embryos only with hydrogen peroxide, he found that these embryos actually finish epibole much earlier when he compared these embryos with the embryos that were treated or incubated in a control media. So if we take into account all these results that I have shown you before, so far we know that actually during early stages of development in zebrafish, NOx enzymes are very active. They are responsible for the formation of ROS. In particular, we think that is hydrogen peroxide that is very active at these stages of development. And somehow ROS are important and are required for epibole to take place in zebrafish development. However, we wanted to know how the ROS actually are participating in the regulation of epibole, and we wanted to know which are the targets of ROS. activity. And for that, we started to look for some usual suspects or potential targets, because work from many different labs that work at these stages of development in zebrafish have found actually that the cytoskeleton of actin and microtubules are very important for the process of puberty. If they interfere with the actin or microtubules, they destabilize these proteins. Puberty is affected. Also, we were very interested in A-cadherin because they have Bakin mutant that was reported when in the group of Nusslein Bolhard did the mutagenesis screening in zebrafish that was reported in 1996. He found that Hapbaked gene, actually this mutant, showed delay in the puberty, mainly of the deep cell layer. Yeah, and this is to some degree similar what we found when we treat the embryos with the general NOx inhibitor. Actually, years later, different groups found that Hapbaked code for E-cadherin. And E-cadherin is an interesting protein because, as you may know, it participates in the formation of addition complexes that allow addition to take place in between cells, neighboring cells. So Mario, well, we wanted to know, and Mario did these experiments. We wanted to know if when we downregulate ROS formation by treatment with this general NOx inhibitor, the pattern of localization of actin filaments and E-cadherin is affected at the enveloping layer cells, but also if we affect the microtubules that are present mostly and they are more easily imaged at the Jolt cell because they show a very large and huge microtubules in this tissue. So Mario actually found that these two different proteins are affected. But I want you to focus mainly on the effect on E-cadherin, because the signal becomes quite dispersed at the enveloping layer cells when we downregulate the formation of ROS. And the localization of E-cadherin can be rescued when we treat the embryos with hydrogen peroxide. So hydrogen peroxide is required for survival, development, and is important for the proper localization of different proteins. And the changes in the pattern of localization of E-cadherin actually suggested to us that cell addition was affected when we downregulate ROS formation. And for that, Mario disgregated embryos that were at sphere stage. And these the cells were put into a tissue culture system that is known as the hanging drop system. So this system is very nice because it just by simple gravity, cells go to the bottom of the media, yeah, of the drop of media. And in that system, when we incubate the embryos in control media, these cells actually interact among each other and form very nice aggregates, compact aggregates after 4 to 8 hours of tissue culture in the system. However, when we treat the cells, these disgregated cells with the general NOx inhibitor at different concentrations, at higher concentrations, the cells start to have problems to aggregate among them, and they still remain dispersed in the media. So they actually have problems to form addition complexes among them. Yeah. So this means, and in general, these results show us that ROS, and it seems that hydrogen peroxide is the ROS that somehow is affecting cell addition by affecting the localization pattern or behavior of E-cadherin, but also is affecting different cytoskeleton components, and these effects are important for a puberty to progress. E-cadering we then started to focus mostly on e-cadering. E-cadering is synthesized as a precursor of 140 kilodalton size. Is processed by furins inside the cells, and these give rise to 120 kilodalton protein that is translocated into the plasma membrane, and there e-gathering then expose the extracellular domains that interact with the neighboring e-gathering proteins that are present in neighboring cells, and this allows the formation of addition complexes among these cells. These addition complexes and addition in general during these stages of development is important to be regulated, to have it shows a very dynamic behavior, because cells at these stages of development use as a substrate for migration other cells, because the embryo at these stages of development doesn't have too much extracellular matrix proteins at those moments. So they have to, in order to be able to migrate the cells, they have to keep the addition behavior very dynamic. And this happens because when the addition complexes are destabilized, the E-gathering can be endocyted by clathrin and dynamine-dependent mechanisms. So once the E-gathering is in the endosomes, it can be recycled back into the plasma membrane. And then usually the recycling occurs at different regions of the cell membrane, and this allowed the additions to be very dynamic. However, some of these endocyted E-gathering also go to the process of degradation through these two different systems in the lysosomes or by the proteasome system. So we think, and well, here I can, Denise Nabel Peraza and Arlene Ramirez Corona performed some Western blot analysis, and as previously reported by other authors, the main protein that is synthesized at that moment or is found by Western blotting is the large 140 kilodalton protein, and the major E-gathering of 120 kilodalton is located at the plasma membrane and here in the enveloping layer cells. We think that when we downregulate the ROS formation by treatment of the embryos with the general NOx inhibitor, this can be affecting the amount of endocytosis or the intensity of the endocytosis that is taking place in the enveloping layer cells. And we think that also because this has been reported by other groups, that when endocytosis is increased, the endocytosis of E-gathering is increased, usually this protein goes to the degradation process. And perhaps this is happening in the cells of the enveloping layer, and that is why we see a downregulation and dispersed pattern in these cells. And this can be recovered by treatment with hydrogen peroxide. But we think also that somehow the recycling process is not completely normal when we downregulate ROS formation. So for that, we ask if we could recover the phenotype if we affect the first step of the endocytosis or the endocytic vesicles formation. And for that, we started to use a dynamine 2 inhibitor, Dinosaur, because dynamine 2 is a large GTPase that actually is important for the separation, for the individualization of early endosomes, because actually the dynamine 2 strangled the neck and then separate the early endosomes from the plasma membrane. And we thought that if we treat zebrafish embryos with Dinosaur, maybe we can force the retention of E-gathering at the plasma membrane, and this might recover the phenotype of or the development of zebrafish embryos that have been treated with this general NOx inhibitor. And that's what Javier Mendez Cruz actually found. And during this his PhD research project, this is a manuscript in preparation. First, he started to verify that we can confirm the effects of general NOx inhibitor. He found that, actually they showed delay in epibole when compared with control embryos. These are lateral and vegetal pole views of embryos by fluorescence microscopy. He again showed that hydrogen peroxide is able to rescue the epibole delay, but also when he treats with dinosaur at this concentration, embryos that are treated also with the general NOx inhibitor, the epibole is rescued, the epibole delay promoted by the ROS downregulation. So, and the rescue, he was able to show that he rescued the enveloping layer epibole and the deep cell epibole. And as expected, Arlene Ramirez Corona, during her master degree thesis research work, she also showed that treatment with dinosaur actually rescued the effects of VAS 2870, because as I showed you before, when we downregulate the formation of ROS in embryos undergoing epibole, the E-gathering is downregulated at the plasma membrane, but also become more dispersed pattern when the embryos are treated with these compounds in the enveloping layer cells. And dinosaur is able to rescue the normal localization pattern of E-gathering, similar to what is found when we treat the embryos with hydrogen peroxide. Also, as I mentioned before, embryos in this culture system, when they are treated with exposed just to the control media, they develop perfectly by 24 hours of development. But when we treat the embryos constantly with the general NOx inhibitor, all the embryos actually died after 24 hours of exposure to this inhibitor. But Arlene Ramirez Corona was showed when she treated the embryos with the very same concentrations of dinosaur. Not only the survival of the embryos is recovered, also the whole complete development is rescued, and these embryos show really no differences, at least at this level of analysis, with the control embryos. Yeah. So far we think that actually ROS perhaps is actually hydrogen peroxide that accumulates at the extracellular space in the embryos that are undergoing epibole. Yeah, and it is required for the normal cycling of E-gathering and for the maintenance of the addition complexes that are formed by E-gathering. And there is a normal process of degradation in these embryos and the cells of the embryos. However, we think that when we affect NOx activity by treating the embryos with this general NOx inhibitor, the ROS are not accumulating anymore at the extracellular space, and somehow this destabilize the addition complexes, and this increase. We think that this is somehow is increasing the endocytosis process and the degradation of the endocyte E-gathering. Yeah. This can be rescued by when we add to the system hydrogen peroxide, but also we can rescue the phenotype when we add dinosaur that inhibits. We think that this is just a partial inhibition of the dynamine 2 activity, because otherwise, if we stop completely the endocytosis process, it has been shown by other groups that this also affects the epibole process. So we still wanted to know if the quotation was increased somehow when we downregulate the activity of NOx enzymes and we downregulate the amount of ROS in the system. And for that, Javier Mendez Cruz actually, he showed again that when we treat the embryos with the general NOx inhibitor, epiboly is delayed. But when he treated the embryos with this proteasome inhibitor, MG132, he found that this proteasome inhibitor actually rescue both the epiboly of both cell layers, the enveloping layer cells and the epiboly of deep cell layer. And its partial rescue is not as spectacular as when we treat the embryos with dinosaur, but this shows that also when we downregulate ROS in these embryos, protein degradation is increased, at least by the proteasome system. Well, in summary, so far, what we know is that NOx are very active during these stages of development, and they are responsible for ROS formation. In particular, it seems to be that hydrogen peroxide is the main ROS that is formed thanks to the activity of NOxes. And hydrogen peroxide is very important for the dynamic behavior of E-cadherin, the recycling process, and also somehow it participates in the regulation of degradation of E-cadherin in these embryos. Also, hydrogen peroxide is important for the maintenance of the cytoskeleton structures, and all these proteins are important for the regulation of the adhesion dynamics of the cells, and this affects cell motility that is fundamental for gastrulation to take place, and in particular of epiboly. But now we know, and we have many additional actually experiments that shows that ROS affect other proteins that are important for the regulation of the dynamics of these proteins and for the regulation of cell motility during gastrulation and epiboly. But this is also another manuscript that is in preparation, but I still don't want to show those results. But what I can say is that we are, thanks to the hard work of Francisco Javier Mendez Cruz, and with the help of Janet Palacios, that is helping us a lot with bioinformatic analysis of the transcriptomes of embryos in which we have downregulated the formation of ROS. They show quite a dramatic changes in their transcriptomes, and these changes in the transcriptome behavior can be fully rescued when we treat the embryos with hydrogen peroxide. Yeah, I also thank the hard work from Brenda Reza Medina and Arlene Ramirez Corona, because they are helping also to annotate the differential expressed genes that we found with these different treatments. Finally, I want to thank all the students and all the people that have been involved during this project. But also I want to thank especially Denise Nabel Peraza and Hilda Lomelí, because together with them, we established the Zebrafish facilities at the Instituto de Biotecnología de la UNAM in Cuernavaca, here in Mexico. I didn't have time to show some of the results that we have together with Dr. Laura Alvarez and Mayra Antunes from the University of Morelos here in Cuernavaca, because they helped us to generate some control experiments important for this project. Also, I want to thank the hard work from the technicians of the lab, Laura Ramirez and Dulce Pacheco, and the funding agencies, Papitunam, with this project number, and CONACYT. And finally, I want to thank you all for your attention, and I hope we can still have time for some questions. Well, thank you very much for your attention.
Oh, hello, Professor Enrique. Thank you very much for this very nice talk. Very informative. We have two questions from the audience. The first one is from Jimmy, and he's from the UNAM Biotechnology Institute, Mexico. He's asking if you will ever have you performed experiment adding H2O2 and dinosaur at the same time? If not, what do you think it would happen?
Well, and it depends on the concentrations of these. Yeah, thank you for the question. I don't know what could happen, but maybe if we down-regulated activity of NOx enzymes, actually maybe it will work much better as a rescue experiment. But that depends quite a lot on the concentrations that we use for doing these experiments. The problem is that hydrogen peroxide is actually can oxidize different molecules in these tissue culture systems. But we, at least for VAS 2870, we have shown that most of these molecules is not truly highly affected by hydrogen peroxide exposure when we perform these rescue experiments. But we should test whether dinosaur can be affected, oxidized by hydrogen peroxide. But yeah, maybe there is a good system to rescue the effects of downregulating NOx activity.
Nice.
Yeah.
The other question we have is from Flavio Zolesi, and he's actually initiated by excellent talk. And I guess you would expect actin dynamics to be important here. Have you looked at it? I'm thinking on experiments in the line of what Jen Hawking showed earlier.
Yeah, we should be affecting, in general, the cytoskeleton. Yeah, not only acting dynamics. Also, as I show you, the microtubules are also affected. We analyzed the microtubules that are formed in the yolk cell because these are more easily analyzable, these microtubules, because they are very large microtubules that are actually oriented in the animal to vegetal pole and in the yolk. But yeah, for sure they should be affected. And it's interesting because also microtubules and some proteins that interact with microtubules and acting themselves, they have in their protein sequence some cysteines that are actually known to be that can be changed their redox state due to the action of hydrogen peroxide. So yeah, that might be important for them. Also, some of the effects that we see in on the zebrafish embryos when we downregulate the ROS formation. Yeah. Thank you. And Kindra, well, is my dog, but she's barking all the time. Sorry for that.
So that was the only two questions we had. So I think we're going to end here. So thank you very, very much, Dr. Enrique, for this presentation and for accepting our invitation to be here. And we hope to see you soon again.
Yeah, well, thank you. And maybe someday we will go back again to Brazil.
Yeah,
very nice.
Please come to Rio.
Okay, yeah, to dance and to, well, no, to do some research, nice research.
Of course.
Well, thank you very much, Magda. It was a pleasure. Yeah.
It was a pleasure for us.
And thank you for your attention.
My name is Mark.
We're going to be right back with our next guest, okay? So stay here. Don't go anywhere. We'll be right back.
I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries in research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
Saúde, direito de todos e dever do Estado. Direito à moradia, ao emprego, à educação, a um meio ambiente saudável, à água potável, ao lazer, à cultura. Direito à voz e à paz. Respeito à diversidade. Saúde para a Fundação Oswaldo Cruz abriga todas essas dimensões. É assim desde 1900. Uma visão integrada da saúde. Na pesquisa, ensino, assistência, informação, comunicação, memória, na promoção da saúde, na inovação, onde permanentemente se pensam respostas para as necessidades da sociedade, onde se faz ciência em defesa da vida. Você pode até não saber, mas carrega a Fiocruz dentro de você. Desenvolvemos e fabricamos vacinas, medicamentos, biofármacos, testes para diagnóstico. Formamos pessoas do nível médio à pós-graduação. Realizamos pesquisas para superar ameaças à saúde, para diminuir riscos ambientais, para prevenir doenças e agravos. Pesquisas que beneficiam crianças, adultos, idosos. Uma instituição pública e estratégica de Estado, integrante do Sistema Único de Saúde, com uma rica história de contribuições à sociedade. Presente de ponta a ponta no Brasil, onde cada trabalhador é um elo forte e ativo. Nela, ciência e saúde cumprem uma função social para o país e o mundo. Pés fincados na tradição, olhos voltados para o futuro. Somos patrimônio da ciência e da saúde, da humanidade, do povo brasileiro.
Situada no estado do Rio Grande do Norte, no Nordeste brasileiro, a Universidade Federal do Rio Grande do Norte está entre as maiores universidades da região. Fundada em 25 de junho de 1958, possui uma área construída de aproximadamente 300 mil metros quadrados, que compõe os 5 campi distribuídos por todo o estado. Tem hoje mais de 40 mil estudantes divididos em mais de 110 cursos de graduação, mais de 90 cursos de especialização, incluindo as residências, 80 cursos de mestrado e 40 de doutorado. Conta com mais de 2.300 professores, dos quais mais de 1.500 com doutorado. São mais de 1.900 projetos de pesquisas desenvolvidos pelos mais de 340 grupos de pesquisa cadastrados no CNPq, mais de 700 projetos de extensão e 4 programas permanentes, além de mais de 3.000 servidores técnicos administrativos. Como uma universidade de ampla atuação, as atividades de ensino, pesquisa e extensão se estendem a todas as áreas: ciências humanas, ciências sociais, ciências exatas e da terra, ciências biológicas, ciências da saúde, tecnologias e artes. Entre as áreas de destaque da UFRN, podemos citar o Instituto do Cérebro, que realiza pesquisas na área de neuroengenharia para o tratamento de doenças vasculares, epilepsia e outros distúrbios neurais. O Instituto Internacional de Física, que promove a troca de conhecimento científico com a comunidade internacional em áreas estratégicas. O Núcleo de Petróleo e Energias Renováveis, que por meio de parceria com a Petrobras, é considerado um dos maiores polos de pesquisa na área, formando mais de 1.000 profissionais em níveis de graduação e pós-graduação. O Instituto de Medicina Tropical, que tem por objetivo o estudo de doenças infecciosas com maior incidência em continentes de clima tropical, como América do Sul, África e Ásia. O Instituto Metrópole Digital, que atua na formação técnica e acadêmica por meio da inclusão social e digital de jovens, desde o ensino básico até a pós-graduação. A Escola de Música da UFRN, que tem por objetivo educar, produzir, disseminar conhecimento e fazer música como forma de contribuir para o desenvolvimento humano, promover a justiça social, a democracia e a cidadania. Nossa escola acolhe mais de 20 grupos e a Orquestra Sinfônica da UFRN, uma das únicas orquestras universitárias do país. O NUPLAN, Núcleo de Pesquisa de Medicamentos e Alimentos, um laboratório oficial destinado à produção de medicamentos para o Sistema Público de Saúde brasileiro. Entre suas atividades, o NUPLAN também oferece suporte acadêmico para a graduação e a pós-graduação, promovendo o ensino, a pesquisa, a extensão e a inovação tecnológica. O Ágora, Instituto de Línguas, Literaturas e Culturas Estrangeiras Modernas, que tem como principal objetivo oferecer cursos de língua estrangeira para estudantes, técnicos administrativos e professores da UFRN, bem como testes de proficiência para possibilitar a mobilidade internacional na universidade. O Ágora também recebe estudantes e pesquisadores das instituições parceiras da UFRN e de outros países, por meio do curso gratuito de português para estrangeiros. Na extensão, o programa de maior abrangência é o Trilhas Potiguares, que leva o conhecimento de professores e alunos aos municípios do interior do Rio Grande do Norte, em ações sociais desenvolvidas em parceria com as prefeituras municipais. Com mais de 20 anos de atuação, o sucesso desse programa já o levou também ao exterior, com a ida de nossas equipes a Moçambique, atuando com as equipes locais. Outra ação de relevo da instituição é a Semana de Ciência, Tecnologia e Cultura da Universidade Federal do Rio Grande do Norte, maior evento de atividades científicas, tecnológicas e culturais do Estado e um dos maiores palcos para a interface da UFRN com a sociedade. Em sua estrutura, a UFRN também dispõe de 3 hospitais universitários, 2 deles na capital e um no interior, 22 bibliotecas, 3 museus, canal de TV digital, rádio, agência de comunicação, editora, núcleo de arte e cultura, núcleo de educação infantil, residência e restaurante universitário. Em seu polo esportivo, disponibiliza campos de futebol e de treinamento oficiais, ginásios, piscinas olímpica e semiolímpicas. Assim, a UFRN busca a excelência e a inovação acadêmica por meio da formação de profissionais capacitados e ações educacionais comprometidas com a produção de conhecimento articulado com a integração social e comunitária, desenvolvendo soluções para os problemas regionais, nacionais e internacionais.
generate, and spread knowledge to form transforming agents. This is the goal of the biggest university of Rio Grande do Sul, State Northern Region, and one of the biggest from the country's southern region. With half a century of history, UPF maintains its roots in educational excellence, research and extension incentive, and the constant search for technological innovation. There are 9 campuses that are distributed around the region and 12 academic units, offering undergraduate, specialization, master's, doctorate, and postdoctorate courses. The infrastructure is a great differentiator. There are clinics, rooms for practical experimental teaching, laboratories, amphitheaters, auditoriums, and libraries with hundreds of thousands of titles available, both in physical and digital format. UPF stands out for the quality of teaching, academic, investigative in nature, the dialogue with the community, the performance in the job market of professionals who have graduated from this institution, and by the qualified teaching staff with teachers who promote the renewal of methodologies for the teaching and learning process. The research incentive in the university generates and disseminates knowledge. The extension reinforces the university's commitment to the community, with projects and programs that, through services provided, approximate academics to social reality and contribute to the sustainable development. Along with a university's social responsibility policy, the institution promotes citizenship, environmental awareness, social inclusion, culture, and development. Major events are held at the university. An example is the National Journey of Literature, one of the greatest literary manifestations in Latin America. The Radio UPF and UPF TV broadcasters with open signal and educational character provide the community with cultural programming focused on local development. Facilitating access to higher education, the university has several lines of credit, scholarships and funding, benefiting more than 70% of students. One of them is the PAE, UPF's own financing program. Every semester, possibilities of exchange are offered to students through agreements with institutions in several countries. Acknowledgements strengthen the greatness of UPF. The university is among the best private institutions in Brazil. Its courses received grades 4 and 5 in the assessments made by the Ministry of Education. The institution has also received in recent years other important awards, such as Social Responsibility, granted by the State Legislative Assembly. The Planalto Medio UPF Scientific Park contributes directly to the development of the region. Connecting innovation, science, and technology, the UPF Parque, with its technology-based incubator, supports and encourages innovative projects and local entrepreneurship. Providing quality of life, UPF offers to the academic community services that favor inclusion and permanence in higher education and the construction of great stories. The university has a community center, a leisure environment to eat, interact, and rest. This is the University of Passo Fundo, an institution that prioritizes ethical and humanistic formation. UPF, knowledge is our nature.
Good afternoon, everyone. So I'm Claudia. I'm a PhD candidate from the Department of Biochemistry at the Medical School of Ribeirão Preto at the University of São Paulo. And now I'm going to introduce you our next presenter, next speaker. She's Magdalena Cardenas. She's a senior research associate at the Human Molecular Genetics Lab at the Institut Pasteur in Montevideo, Uruguay. And she currently investigates the processes related to kidney diseases by using the zebrafish models. Okay, so I would like you all to welcome Magdalena. And I would like to remind you all that you can send all your questions on our comments on right below the presentation, so they can be answered later. Okay, so welcome, Magdalena. You may begin.
Okay, hi, Claudia. Thank you very much. I will start my presentation. Just let me know if everything goes okay. You can see everything, it's okay? Okay, so I will start. Thank you very much to the organizers. My idea in this presentation is to share with you some data from one of the projects that I work during my postdoc. Since I don't know how familiar you are with cilia biology, I will do a very brief introduction related with cilia, just to be tuned in the same thing. So what we know and what are cilia? Cilia are microtubule-based cellular organelles that protrude from the surface of the cells, and their presence has been described in almost all cell types. The main body, let me put here a laser point, okay. The main body of cilia is a microtubule structure composed of alpha and beta tubulin that they distribute in 9 doublets that they distribute the periphery of the cilia. Depending on the structure, if they present a central pair of microtubules, they can be motile or non-motile. So motile cilia present a 9+2 structure, and non-motile cilia lack the central pair. Motile cilia also present dynein arms that allowed the movement of the cilia. Also, the axoneme is possess a membrane that is a plasma membrane, a membrane that is continuing with the plasma membrane of the cells, and the cilia is in the base is root to a basal body that is a modified centriole that come from the centrosome, and the cells can present motile cilia, as I said, or unmotile cilia or primary cilia that can be 1 per cell in the case of non-motile cilia, or some cells like multiciliated cells can present a dozen of cilia in the apical surface. Cilia present a special transport system that allowed things go in and go out from the cilia. And what we know actually from the function of the cilia is that they are central hubs of signaling pathways. They are sensory and mechanical sensory organelles that process signal from the outside and from the environment and take all that information inside the cells. So for a long time, it was really difficult to understand the role of this organelle, but nowadays it's a very important organelle that has been studied in a lot of animal system and in human. And the relevance that this that cilia has is evidence in the embryonic development and in the physiology of different tissues and organs. And it has been shown that the malfunction of cilia is the basis of a group of human genetic diseases that are called ciliopathies. Nowadays, there are at least 35 different ciliopathies described that the prevalent phenotypes that they include are polycystic kidney disease, retinal degeneration, obesity, skeletal malformation, and brain anomalies. And as you may see here, almost all the tissues and organs are affected when there is a malfunction of cilia. So one of the zebrafish has been one of the most popular animal models used to study cilia biology. And that's not only because of the advantage that this model presents, that for sure you know, also is because they present several ciliated organelles that are easy to study and to do imaging and to study how cilia is there. So I will just very briefly introduce you some of the ciliated organelles that are more studied in the case of zebrafish for cilia studies. One is, as you may know, is Kapfer vesicle, that is a transient organ that is related with the establishment of left-right asymmetry in the embryo, and they possess very long cilia that they are easily to stain and analyze. Also, the pronephric ducts, the epithelial cells that line the pronephros in the zebrafish present cilia. In this case, they are multicilia, and cells can have one cilia, or they are also multiciliated cells, where cilia distribute to the lumen of the tubules, where they sense the flow that goes inside the lumens. Also, the olfactory pit, the nose, it's a very study organelle because it's located outside in the embryo, so it's easy to study. The cilia of the spinal canal, also they are a good model to study cilia. And since the photoreceptor present the outer segment of the photoreceptor is a modified cilium, also they had been studied for understand how cilia and proteins related with cilia biology are working there. So I will switch now to show you one of the projects that I worked during my postdoc at Ian Drummond's lab at Harvard Medical School in Boston. In this project, we studied the centrosomal protein CYP290, which has been related to several ciliopathies, and it has been difficult to establish clear correlation between mutations and phenotypes. So, as I said, CYP290 is a centrosomal protein. It's a very big protein, a big gene, so it's difficult to work with. In zebrafish, the gene has 52 exons. The protein has multiple domains, at least 13 coiled coils. And what we know about CYP290, it localizes in centrosomes and also in basal body and in cilia transition zone, where it acts as a hub where it connects different macromolecular complex related with transport in and outside the cilia. Also, it has a role in the microtubule organization, migration of the centriole, mother centriole to the apical surface, and trafficking to the centrosome. Also, it has a key role in the formation and maintenance of the outer segment of the photoreceptors. But the role that it has in ciliogenesis is not completely understood because depending on the animal model that is studied, there are different results, and was not so easy to understand its role there, and also because there are tissue-specific roles of this protein. So one of the important particularity of this gene, as I said, is an important gene because it has been described as a causal gene in at least 6 different ciliopathies. that the interesting thing is that they vary greatly in the spectrum of severity and the affected organs. For example, mutation in this gene in CYP290 are associated with 30% of cases of level congenital amaurosis, which is characterized by childhood blindness due to retinal degeneration, with no other associated systemic disease. But on the other hand, also is a causal gene of Meckel-Gruber syndrome, where multiple organs are affected and is embryonic lethal. So today, 130 mutation in this gene has been associated with human diseases, and most of the mutations that had been identified are nonsense mutation or frame shifts in the open reading frame that makes a premature termination pattern appear. So it has, as I said, it has been difficult to determine a clear genotype-phenotype association for CYP290 from the animal models or even from patients that show CYP290 mutations. So here, very briefly, just to show you that there are a lot of animals, several animal models with mutation in CYP290 that have been developed in mice, in cats, zebrafish, C. elegans. I will not go in detail with this, but the problem of all of this is that they show a variety of phenotypes or phenotypes that appear at different times in the development. So that's what makes different show a correlation between phenotypes and genotypes. So different possible explanations had been proposed to explain this pleiotropy. One has been that maybe modifier genes are making this difference between the phenotypes that the patients or the animal models show. But this explanation can't be explaining all the different, not all the cases. So also it had been proposed and had been shown also in some cases and not in all, that a genetic mechanism of basal exon skipping or nonsense associated alternative splicing can be removing the mutated exon. And in that way, some fully functional CYP290 protein is still expressed. So this has been proposed, but not all the patients can be explained. Not all the phenotypes of the patient can be explained by these mechanisms. Since some patients show the same levels of alternative splice messenger as the control. And the third possible explanation to explain CYP290 pleiotropy has been possibility of that genetic compensation mechanism is acting in this case. As you may know, in the last year, there had been shown that deleterious mutations are not gene knockdowns induce a genetic compensation mechanisms to counteract the genetic loss. And the presence of these mechanisms has been shown in in several models. So we decided to focus and analyze this possibility and see if it can explain the pleiotropy that is seen in CYP290. So our idea was compare cilia-associated phenotype in an acute zebrafish morpholino knockdown with a stable CRISPR-Cas9 CYP290 mutant zebrafish model. So the first thing that we did was to develop two models in zebrafish of CYP290 inhibition. One was an acute model where we used a morpholino to knock down CYP290 expression, and also we did a CRISPR-Cas9 mutant line. The morpholino that we used was against exon 25 that makes the retention of an intron, change in the open reading frame, and the presence of a premature stop codon. In the case of the mutant line, it's a 10 base pair deletion, and also it caused the frame sheet in the open reading frame and a stop codon in exon 16. So analyzing the phenotypes that we observed, clearly the disruption of CEP290 caused ciliopathy in fish. In the case of the morphan, they show a clear cilia-related phenotype, such as body curvature, otolith defects, the development of cysts in the kidneys, left-right asymmetry defects, and the shortening of cilia. We also developed 2 antibodies against zebrafish CEP290 against the amino terminal and the C-terminal domains. And using those antibodies, we showed that in our model, there is no expression of the CEP290 protein. But we saw some difference in the phenotypes between the acute model and the mutant line. The general morphology of the fish is the phenotype is milder. The body curvature is not so curved. And also we saw a penetrance of the phenotype that was clutch dependent. It was between 20 or 50%. There was no kidney cyst, no otolith defects, and no left-right asymmetry defects. So we decided to look what was happening with cilia in these 2 models, and for that we used the antibody that we produced, and we analyzed different tissues in both models. For example, we analyzed the pronephroid ducts and analyzed cilia, the olfactory plateau, the eye, and the Kupffer vesicle cilia. What we saw first, using this antibody, we couldn't detect immunoreactivity of CEP290, what validate our model. And also we saw some difference between both models. We saw in the pronephros, this is the morphan, this is the mutant. We don't see any difference in the structure of the pronephros. The olfactory plateau, it can be some structural changes, but was not evident or not very easy to quantify. But when we look at the eye, we saw that there was a modification of retinal lamination also in the morphans and in the mutants. And when we analyzed the length of the cilia of the Kupffer vesicle cells, we saw that the morphans show a shortening of cilia length, but not that shortening was not seen in the mutants. So there is the loss of CEP290 in an acute or a long-term loss model differentially affects at least Kupffer vesicle cilia. So since the role of CEP290 described in the structure and maintenance of the photoreceptor outer segment length, and that we saw this modification in the retinal lamination, we decided to see in more detail that. So we analyze the structure of the outer segment of photoreceptor. For this, we use an internal membrane marker that is called body PTR. And we analyze in control, in the morphans, in the mutants, and in the heterozygous that they don't show phenotype. So what we saw is that in the case of the morphan, there was a reduction of the length of the outer segment of the photoreceptor. Even very few of the photoreceptors show outer segments. In the case of the mutants, we saw a disorganization of the outer segment, but there was an outer segment there. And what we saw is that by electron microscopy, we detect an accumulation of vesicles in the base of connecting cilium of the photoreceptors. The mutant animal reach adulthood. They are able to reproduce, although with some difficulty, but they only present spinal scoliosis. So we analyzed in the adult retina what was happening, and what we saw is that the alteration of the photoreceptor that we saw at larvae stage persists until adulthood. So we decided then to analyze if what was happening with these fish, because one observation that we had that was the more striking, that was that at 3 days post-fertilization, the mutant embryos, larvae, present a modification in the axis, a curvature in the axis of the body. But that curvature disappeared at 7 days post-fertilization. It's like it recovered to a wild-type phenotype. So that was an interesting observation. So we decided to explore why that was happening. As I mentioned, this is an adult mutant that they show a strong spinal scoliosis. So we decided to focus to understand how this is recover, how they recover to a wild-type phenotype. So the first thing that we did was to analyze if some basal exon skipping or non-cell mediated alternative splicing was happening around the mutated exon. So we designed primers around 10 exons around the mutated exons and analyzed the size of the applicants, and we couldn't detect any difference between the wild type and the mutant maternal zygotic mutants. So that means that there was not the mutated exon was still there and was not being exon skipping or alternative splice. So since the mutation is a 10 base per deletion that generates premature stop codon, we compare the messenger levels in the mutants with the controls. So we used specific primers that amplified the wild type exon 16, and we were unable to amplify the wild type messenger in the maternal zygotic mutant samples. In contrast, we detected a fold change of a lot 1,000 in eye samples and a fold change of 200 in kidney samples of the mutant respect to the control of the mutated mRNA. So also we decided to analyze using primers against the N-terminal, the middle part of the messenger, the RNA, and the C-terminal domain of the messenger, and see and compare between the mutant and controls and see what happened with the levels of the messenger. In this case, although we show there is a premature stop codon in the mutated mRNA, we didn't see that the messenger is being degraded by nonsense mediated decay mechanisms. So that we couldn't detect, although the premature stop codon is far enough from the last exon-exon junction, we couldn't detect NMD degradation of the messenger. So we decided to analyze if genetic compensation is responsible of these milder phenotypes in the mutants, and if it's the reason why phenotypes are recovered in the mutant. So just one thing important to mention is that up to today, no parallel gene of CEP290 has been identified in zebrafish, and also there's no duplication of the gene. That's something important. So what we did was an RNA-seq analysis of the transcriptome of the mutants, the morphans, the mutant, the morphans, and the wild-type embryos, and compare between them. Which was our hypothesis? Our hypothesis was that if the lack of CEP290 is compensated by the expression of other genes, we should observe an upregulation of those genes only in the mutants and not in the wild type or in the morphans embryos. So from the genes that we obtain from that analysis, we filter with a cilia proteome database, and we decided to focus in some genes that are cilia related or are related with the function of CEP290. From those genes, we decided to focus in R3, R13B, and ANC119B. What we know very briefly about these genes or these proteins, R3 is a small GTPase that localize to cilia and is involved in the transport of lipidated proteins to the cilia. R13B is a small GTPases that localize to the cilia membrane, where it regulates the localization of membrane proteins and participates in the stability of IFT complex, and also interact with R3. And in the case of ANC119B, it's a cargo adapter that binds myristoyl-related proteins. It localizes in the transition zone and interacts with R3. So what we did first, these are the results from our RNA-seq from maternal zygotic mutants. And also the first thing is that we check by real-time PCR in samples from eyes from adult mutants, maternal zygotic mutants, and we confirm the upregulation of these 3 genes. Also, we check that the morphans, they don't show an upregulation of these 3 genes. And some interesting thing is that in the zygotic mutants of CEP290, we couldn't detect an upregulation of the genes, meaning that the presence of the CEP290 maternal messenger is enough to avoid or not to induce this upregulation, this genetic compensation. Also, we check if other cilia mutant lines also show an upregulation of these genes, and we couldn't detect that, meaning that this is not because of a cilia defect and is specific for the loss of CEP290. So we also analyzed some samples from patients that Schubert patients that it's a ciliopathy where CEP290 is mutated. CEP290 is a causal gene in this syndrome. It's a ciliopathy. So we analyze urine-derived renal epithelial cells from these patients and show that ANC119B also is upregulated. We couldn't detect an upregulation of R13B and R3. But interestingly, when we analyze adult kidney samples from our mutant line, we also detect only an upregulation of ANC119B, meaning that there is indicating that there is some tissue-specific regulatory process of this genetic compensation. So we thought, okay, if the upregulation of these genes are compensating the loss of CEP290 in the mutants, the upregulation of these genes should rescue the morphan phenotypes. So to do this and to confirm this, we decided to focus in 3 phenotypes that we were able to measure and quantified. So we decided to focus in the percentage of animal with body curvature, in the length of Kupffer vesicle cilia, and in the length of the photoreceptor outer segments. So what we did, we clone and express the mRNAs of the zebrafish R3, R13B, and ANC119B, and injected them in embryos where also the morpholino against CEP290 was being previously injected. And first, we observed an increase of almost 50% of embryos with straight bodies, like wild type compared with the morphant embryos in which the messenger were not injected. On the other hand, when we analyze the length of Kapfer vesicle cilia and do the rescue of the length, we decided to inject the three genes separately and analyze, and we were able to show that the upregulation of these three genes in the morphants are able to rescue the shortening of Kapfer vesicle cilia that is seen in the morphant's embryo. So then we analyze the what happened with the outer segment length of the photoreceptors. So we did the same experiment. We injected the 3 RNAs of these genes in the morphant embryos and analyzed the length of the outer segments photoreceptor. And we showed that the 3 genes are able to rescue the phenotype. So as conclusion of this work, we show that the loss of CEP290 in zebrafish affects the formation or maintenance of the outer segment of photoreceptor, most likely by mediating membrane vesicle delivery to the connecting cilium. Also, we show that acute CEP290 knockdown produce stronger ciliopathy phenotypes than the CRISPR genetic mutation. That genetic mutation in zebrafish CEP290 induces an upregulation of genes that encodes proteins involved in cilia membrane transport, suggesting genetic compensation mechanisms in the CEP290 mutants. Urine derivative epithelial cells from human Schubert CEP290 patients also show an upregulation of ANC119B, suggesting that this genetic compensation may be a conserved mechanism that contribute to the genotype-phenotype variation that we observe in patients with the CEP290 mutations. And we also show that if we overexpress these 3 cilia-related GTPases, chaperones can rescue defects in cilia at the Kapfer vesicle and the outer segment of photoreceptors that we see in the morphants. So just to finish, I'm perfect in time. I want to acknowledge all the people from Ian Drummond's lab that participate in this project, and also all our collaborators in these projects. I also want to thank all the people from the Human Molecular Genetic Labs where I'm actually working. So thank you very much for your attention, and I will be very happy to take any questions.
Thank you very much, Dr. Magdalena. We have a question here from Flavio. Okay, just a minute. It's going to be on the screen. Yes. So, is it somehow unexpected that the maternal zygotic mutants do not show gene compensation? Any ideas? Do you know if this was found for other genes?
We think one of the proposed mechanisms for how is induced we don't have the molecular explanation of how is occurring the genetic compensation in our model. What is known actually, and it has been shown with some genes, that the degradation of the messenger is what is inducing the genetic compensation. Those fragments of the messenger, it seems that they go back to the nucleus and they are in a certain way activating the regulation, modifying the regulation of those compensating genes. In our case, and there has been other reports, we couldn't see a degradation of our mutated mRNA. So in our case, we don't know. it's something that is not completely known, how is the molecular mechanism. So, but if that is the case, that the degradation of the messenger is what induces the genetic compensation, maybe in the maternal maternal zygotic mutant do not show. I'm thinking what is he said. Maybe in the, because one interesting thing is that in the zygotic mutant, we couldn't detect the genetic compensation. So we don't know. I think I answered the question. Maybe it's that, but in our case, we couldn't see the degradation of the messenger.
All right. So I would like to thank you again, Magdalena, for your amazing presentation. I'm sure everybody enjoyed it. Okay. So I would like to invite everybody to come back again tomorrow live on our YouTube channel, the ICB-USP from the Institute of Biomedical Sciences. So tomorrow we're going to be live again. And thank you very much for coming. Thank you all for attending, and see you tomorrow.
Thank you.
Thank you. Bye-bye.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries in research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.